Thank you so much. I really appreciate the Walleye Tank for allowing me the opportunity to discuss a startup that I co-founded called Retham Technologies. Retham in my native language of Tamil means blood. Um, the problem that I study is a condition called heparin-induced thrombocytopenia. All of you may have heard of heparin, a very commonly used anticoagulant that's given to millions of patients every year. The problem is a subset of treated patients develop signs and symptoms of this severe reaction to heparin called HIT. In some cases, there are clots that form, and in many of the patients who clot, they have dangerous or adverse outcomes such as strokes, cardiac events, and amputations. The diagnosis of HIT is highly complex. It involves two different steps. The first step is a simple assay, an ELISA assay that's done in the hospital, but the problem with this assay is that it is very high false positives, such that for every 10 positive results, 5 are falsely positive. And so often a second assay is sent out to one of 5 reference labs in the United States that are much more expensive, and results coming back in about a week. The problem in the interim for physicians like me is to struggle with how to treat the patient. There are problems if we ignore HIT, and there are problems if we assume that someone's got HIT when they do not have. And this is a problem that has been well recognized by hematologists and transfusion medicine practitioners for some decades. More recently, based on some basic science work in my lab, we made the discovery that if we add a simple protein that can be made in large amounts called PF4 to platelets that Retham has stabilized using proprietary techniques and add a blood sample, this reaction will cause a positive or negative result using a couple of different platforms that we're considering. The first is an LFA, or a lateral flow assay, which the most common LFA you may be familiar with is a urine pregnancy stick. You add a sample and you get a result. The second one, which is a little bit more hands-on, but this technology being much more accurate, is an ELISA assay, which would take 2 to 3 hours. Now, why do we need to solve this problem now? It is because more than 5 patients die of it every day. I'm sorry, am I out of time?
Yes. Sorry.
Okay, sorry about that, but I'll stop.
Very exciting technology. Yeah. So judges, could I request you to turn on your camera and mics?
Maybe I'll start and ask, would you care to finish your pitch describing the technology, the solution?
Sure. So I just have, I think, this slide and that, and one other one. So the cost of HIT or high, more than 5 patients dead every day, $80,000 versus 20, and a lot of liabilities to hospitals. I've provided an example here. We have a team of individuals. We have a part-time CEO on the left. I serve as Chief Science Officer. A co-inventor is the Director of R&D, and we recently hired in the Patent Counsel. Our technology that I showed you in a previous slide is patented with 2 issued patents in the United States, 1 in Japan. Patents are pending in the EU and Canada, and we've published peer-reviewed data in a number of top-tier journals, including Blood and Chest. So at this point where we are, our development of this prototype that I've pictured down on the bottom left of the slide, we've just completed a seed round of about $300,000 from accredited in-state investors in Wisconsin, which is where I'm from. That's where I joined the Mayo Clinic recently, as mentioned by the host. And we're really hoping that I could get connected with the startup community here in Minnesota, because in about a 6 months or a 1-year period, we'll likely be wanting to go in for a Series A, hiring a full-time CEO, et cetera, to move this product forward.
Sure. I guess I'm surprised at the high level of false results that you're getting. I'm assuming that's a PF4 ELISA. I guess I don't fully understand that. But the concern is that the LFA is going to probably be less precise than a traditional ELISA. Do you know the levels of PF4 in these patients? Are they relatively high, or are we trying to detect really low levels?
So the levels of PF4 we've tested in many patients does not appear to be the issue. It's the PF4 binding to substances on the patient's platelet surface that appears to cause pathology. So we, for example, just published a paper in Blood showing that the false positive rate of the ELISA is 50%. So the PPV, the positive predictive value, is about 50%. And in the same study, we used this technology using PF4 platelets in a sample, and it appears far superior. We have not created the LFA prototype, however, so the concerns you raised, we will have to empirically test that. But we have tested it in the ELISA format, and it appears based on a sample size of about 50 patients, select patients that sort of stress the system, that the accuracy appears to be 100%.
How many kits would you presume you'd be selling in a year, and then in about 5 years?
Sure. The total available market, we have accurate estimates in the United States. We expect this to replace the ELISA market, or compete, I should say, with the ELISA market. It's 1 million tests in the US. Our estimates outside the US are somewhat limited. We estimate about 300,000 in Europe, and we're not sure about other areas. We did a lot of discussion regarding sort of pricing, and we sort of, what we ended up, again, these are rough ranges based on our discussions with people who have more expertise than us. We believe the LFA, given its easy use, could possibly be sold even at maybe $150 a test. Given that currently there's significant expense involved with the two-step approach. In fact, one of the previous institutions I was part of, we generated very high significant revenue with the test number 2 listed here, because there are so few labs that actually are able to perform this test.